Okay, let's dive right into this explainer and uncover exactly how the hantavirus acts like a microscopic master thief, pulling off a highly sophisticated heist right inside the human body. We're going to break down the exact mechanics of how this virus breaks in, takes over, and literally turns our own biology against us. To understand this biological break-in, we'll follow the virus step by step. Our agenda for today: number 1, meet the invader. Number 2, the target. Number 3, the break-in. Number 4, the escape. Number 5, the host hijack. And finally, number 6, the big picture. So, section 1, meet the microscopic invader. First up, we need to examine the anatomy of our culprit to see the specialized tools it brings to the job. I mean, every good thief has a toolkit, right? And this virus is definitely no exception. When we look at the surface of the virus, it's basically a spherical lipid envelope encasing its genetic material. But what's really important is that the envelope is decorated with 2 critical glycoproteins, labeled as GN and GC. You can think of these 2 proteins as the ultimate, highly specialized lockpicks. These 2 proteins are absolutely crucial to the heist. GN and GC are the only viral proteins exposed on the outside of the virus. Without them, the virus is completely harmless. It literally cannot attach to or penetrate a human cell. They work together as a team. One handles the initial approach, and the other handles the violent breach. Moving on to section 2, targeting the blood vessels. Now, every master thief needs a high-value target. And for the hantavirus, it's a very specific type of human cell. It doesn't just attack randomly. It knows exactly what it's looking for. The crucial point here is that this virus specifically seeks out and attacks endothelial cells. If you're wondering what those are, just think of endothelial cells as the delicate, microscopic wallpaper lining the inside of your veins, arteries, and capillaries. By targeting these specific cells, the virus turns our own circulatory system into the scene of the crime. Which brings us to section 3, the cellular break-in. Let's move to the main event and see exactly how this microscopic invader breaches the cell's outer defenses using a process that is, honestly, as elegant as it is terrifying. The invasion happens in 4 chronological phases. It starts with attachment on the outside, followed by internalization, which is where the cell is tricked into swallowing the virus. Then comes trafficking deeper into the cell, and it ends with a violent membrane fusion to release its payload. In step 1, the gliadin protein acts like a key, feeling around the outside of the endothelial cell until it finds specific doorknobs. In scientific terms, these doorknobs are receptors like beta-3 integrins or PCDH1. Once gliadin locks onto these receptors, the heist is officially underway. But here is the really fascinating part. Instead of breaking through the wall directly, the virus just knocks on the door and tricks the cell into initiating a process called endocytosis. The cell membrane essentially acts like a trapdoor, folding inward and swallowing the virus alive into a cellular holding room called an endosome. The cell thinks it's captured the virus, but actually, the virus is exactly where it wants to be. All right, section 4, the acidic escape route. Being trapped inside that endosomal holding room isn't the end of the heist. Let's see how the virus uses the cell's own security systems to stage a massive prison break. Normally, the cell pumps acid into this endosome bubble to digest and destroy whatever it just swallowed. But the hantavirus is a step ahead. It actually uses that exact low pH, highly acidic environment as the chemical trigger for its escape. And surprisingly enough, research shows the virus absolutely requires the cell's own cholesterol to successfully pull off this next explosive step. The way the GLAC protein physically transforms is wild. The acid causes the GLAC fusion protein to violently snap, kind of like a spring-loaded harpoon. It shapeshifts, throwing a fusion loop that literally pierces the wall of the endosome, grabbing hold of the host membrane and forcing the viral envelope to fuse with it. Basically, the vault door is blown wide open. Section 5, hijacking the host factory. With the vault successfully breached and the viral membrane fused with the cell's interior, the virus unleashes its payload and takes total control of the host. The viral RNA spills out into the cell's cytoplasm, and it performs an act of literal microscopic identity theft through a mechanism called cap snatching. The virus's enzymes actually snip the protective caps off the host cell's own messenger RNA and use them to disguise its own viral commands. It tricks the human cell's ribosomes into reading the virus's blueprints instead of our own. Suddenly, the host cell is entirely hijacked. It's forced to build new GN and glycoproteins and replicate the viral RNA. These new parts are sent to the cell's shipping center, the Golgi apparatus, where fresh viral particles are assembled, packaged, and then pushed out of the cell via exocytosis to infect neighboring blood vessels. The victim has literally become the factory. That takes us to Section 6, why this matters. Now that we understand the microscopic mechanics of this incredible cellular break-in, we really need to look at the macro-level damage this heist causes to the human body. So what happens when blood vessel cells are completely hijacked? It's one thing to look at a single cell being overtaken, but what happens when billions of these microscopic break-ins happen simultaneously across your entire circulatory system? Well, the results are, unfortunately, devastating. Because those endothelial cells, that delicate wallpaper lining the blood vessels, are damaged and dysfunctional. They start to fail at their primary job, keeping fluid contained. The blood vessels become incredibly leaky. Depending on the specific strain of the virus, blood plasma leaks rapidly into the lungs, causing life-threatening pulmonary edema, or into the kidneys, causing severe hemorrhagic fever. To really put this heist into perspective, let's compare how hantavirus operates next to SARS-CoV-2. While the virus that causes COVID-19 is highly flexible and can easily fuse its spike protein directly at the cell surface to get inside quickly, hantavirus must take the complicated route. It absolutely relies on being swallowed into that acidic endosome to survive and replicate. It's a much slower, stricter pathway, but man, is it incredibly effective once it's inside. What's really wild to think about is that this incredibly complex cellular heist, the lockpicks, the trapdoor, the acid trigger, was never actually meant for us. In their natural rodent hosts, like deer mice, hantaviruses cause a persistent but completely asymptomatic infection. The rodents don't get sick at all. Human infection is just a biological accident, a deadly spillover event that happens when we breathe in aerosolized rodent dust. Which brings us to the ultimate question. Now that we've mapped out the exact mechanics of this microscopic break-in, from the JAN attachment to the Glick harpoon fusion, we know exactly where the virus's vulnerabilities are. If it completely relies on human cholesterol and an acidic endosome to escape, perhaps the key to a future cure lies in simply changing the locks, or maybe jamming that trapdoor shut forever. Definitely something to think about. Thanks for joining me on this explainer, and keep your curiosity sharp.